I'm giving this presentation in name of Patricio, who could not make it here today, and he is a PhD student in Ghent University, and he's performing metagenome analysis of communities that are degrading pesticides. So pesticides have been used for quite a while in agriculture. In Europe now, some of these pesticides have been forbidden. So like Lineron, one of the pesticides that has been used for a long time, hasn't been used for more than 20 years, but the pesticides are still in the ground. They have been leaching to the groundwater. That's why in bioform purification systems that are used to decontaminate groundwater, you still find communities that are able to degrade the pesticides that were previously present in the soil, like Lineron. Now, this metabolic capacity is still present, although after even 20 years, these soils have not seen any pesticides anymore. So there is definitely some genetic memory. And we wondered here whether these communities could be adapted easily towards degrading pesticides that are related to Lineron, but that they didn't have seen before in the past, so that maybe they could retune their metabolic capacity to degrade novel pesticides. And so to study this, we performed an evolution experiment in which we took 3 types of materials, so bioform purification systems, one which was enriched with Lineron again for a few years so that the communities in these bioform purification systems were adapted again to Lineron. One BPS minus system, so this is the system in which no extra Lineron was added, so that means that these communities haven't seen Lineron for more than 20 years. And then a homemade BPS system, which contains bacteria soil that has never seen any pesticides in the past. And so to each of those BPS systems, we now added as a control milk water or 2 novel pesticides, CLP and CLT. We then assume that the BPS systems that are being treated with milk water still are able to degrade Lineron because they have a genetic memory in the soil, and we wanted to assess whether they were also able to degrade any of these novel pesticides. This is here illustrated in a mineralization assay, which assesses to what extent different pesticides are degraded. So for each of the BPS systems, we let them evolve for about 2 years. And at several time points indicated here, we took samples. Time point 0 is the time point at which the experiment was started. Time point 1 is after 4 months, time point 2 after 17 months, and time point 3 after 24 months. And then we assessed at each time point whether there was a mineralization capacity of the community, or saying otherwise, whether the community was able to adapt to the novel pesticide and metabolize it. And that was the case for Lineron in almost all cases, also in the material that had never seen the pesticide before, although to a very low degree. For the chloropropam, this was also the case. For the chlorotoluron, the capacity was there, but only in few samples and not at the same capacity as the degradation capacity of Lineron and chloropropam. Now, to see which communities were present, in each of those samples, we performed a metagenome analysis, but it's a metagenome that was enriched by DNA isotope probing. That means that the pesticides were labeled with radioactive carbon, C13 carbon, so that if the bacteria in the community degrade the pesticide, then they contain this labeled carbon in their genome, and then their genomes and their DNA can be separated from the rest of the species in the community. So we enrich them in situ without having a cultivation bias. And then we performed on all of those samples metagenome sequencing. We performed all the necessary pre-processing steps, so assembly, binning with different, and we compared different types of methodologies. And then eventually we went to the step of taxonomic profiling. Also there we use different approaches, but what I show you here is the result of plants where you can see that in the different samples, the species that were detected. So in all the samples of the first groups, which I indicate here, these are the ones that were treated with lineron, that degraded lineron or CLP. These seem all to be enriched in either Ramlibacter or Variovorax or any combination thereof. So these are the species that were previously known to be involved in the degradation of lineron. So that means that even in the samples that haven't seen lineron for quite a long time, there is still degrading capacity of lineron, but also an almost immediate capacity of degrading CLP. So probably the same metabolic capacities, same strains are also responsible for degrading CLP. For CLT, the situation is different. Here, the species, it is much less clear which species are really abundantly present. So there, the community seems to be more heterogeneous, and it's less clear which species is really degrading. So from this, we can now already summarize that lineron degradation is mediated by species that were already reported before, that the same species tend to be responsible for the degradation of CLT, but that for CLP, we still need to find out which species contain the degradation capacity and which genes and pathways are responsible. I thank you for your attention.